So chapter 7 is going to focus on the specific structure of the cell membrane and how it's permeable or not permeable to allow certain items in and out of the cell. The plasma membrane is made mostly of phospholipids, which, if you remember back, are part of the group called lipids. Lipids are hydrophobic, but in the case of phospholipids, they tend to have a hydrophobic region and a hydrophilic region, which makes them amphipathic. So amphi means both. And because they have this polar and non-polar region, what happens when you put them in water, which of course is polar, they arrange themselves into a bilayer, where you have the hydrophilic polar heads are both facing the water, and then you have this hydrophobic non-polar tails, which are going to face each other. So inside of here, there is no water. The other big characteristic of the plasma membrane is that it is selectively permeable. So permeable means that you would let something through, but it is selective. So some things are going to be able to cross it, and some things are not. So when we discuss the plasma membrane, we talk about it in terms of what's called a fluid mosaic model. And again, this is a model, so this is a approximation of what the membrane is to help you conceptualize what it is. So let's break it down. The first word here is fluid. So think about it for a moment. What are some things that you know of that are fluids? What does fluid mean? So all liquids and gases are fluids. Fluids are going to be substances that flow. So if you look at this drawing, it's drawn very kind of bumpy and flowing. So when you think of fluid, it means that things are constantly moving or changing. So these proteins are not just randomly distributed, they actually move sideways. So these proteins can typically move sideways through the plasma membrane to get to the regions where they're needed. The lipids, so the phospholipids and the cholesterol molecules, can also move sideways throughout the plasma membrane. It would be very, very rare for a phospholipid to flip over to the other side of the membrane, because then a polar region would have to move through a non-polar region. Instead, what they do is they kind of just move sideways and they switch places next to each other. The membrane lipids and these proteins are always constantly moving. This was seen in an experiment with Fry and Eden. You don't have to remember the scientists, but this is a good illustration of the fluid property of the membrane. So they took a mouse cell and a human cell, which they dyed with different fluorescent dyes here, so you can see that the mouse cell proteins are pink and the human cell proteins are purple. And when they merged these two membranes, if the cell was not fluid, you would expect to see a cell where this half is pink and this half is purple. And at the very beginning, that is what it looked like. But over time, and it only took an hour, you end up with this mixture of pink and purple proteins, showing that the proteins actually moved sideways through fluid membrane. Here you can see a short video of the fluidity of the membrane, and it moves just like water, and the red and white on the outside and the inside are water. The yellow is the polar heads of the phospholipids, and then the green is the non-polar region. Notice again that there is no water here in the areas that are just green, because those are hydrophobic and thus are going to repel water. So in order for a membrane to be functional, it has to maintain this fluidity. If a membrane becomes too solid, it becomes brittle and it can actually break apart, which can destroy the cell. Phospholipids that have unsaturated fatty acid tails tend to create space in between each other, which is going to make something more fluid. Phospholipids that have saturated hydrocarbon tails tend to be easier and tightly packed together, so this is going to become more viscous. Viscous means it doesn't flow very well. I think syrup or honey are fairly viscous. So one component of animal cells is we have cholesterol in the membrane, and this cholesterol actually functions to help maintain the fluidity of our cells at a range of different temperatures. So in an animal cell, the phospholipids are spaced apart using these cholesterol spacers. So when the temperature is warm, it actually prevents the phospholipids from becoming too fluid or moving too far apart. So it kind of creates these little bookends to keep all of those phospholipids in place. And so the cholesterol can kind of limit the movement of phospholipids at a very high temperature. When it's very cold, molecules tend to slow down and the fluidity goes away. And so because those cholesterols create spacers to keep space between these neighboring phospholipids, they help to maintain the fluidity even though the temperature is lower. So cholesterol in the membrane is really important for maintaining the structure and therefore the function of the membrane. Different animals have different lipid composition of cell membranes, and those different compositions can be adaptations to environmental conditions. In this picture, you can see an ice fish. So in this image, you can see an ice fish, and they have different lipids than you and I would have because they live at an extremely different temperature. So natural selection has worked on their cells to select for different compositions of lipids to allow them to maintain the fluidity in their cell membranes at very, very cold temperatures. So the other half of the model is that it is a mosaic, so a fluid mosaic. And if you think about what is a mosaic, you probably think of a bunch of different colorful smashed tiles that are rearranged into a form a picture. And that's exactly what we're looking at. The membrane mosaic is the proteins that are embedded in the membrane. Each protein can have a different type of function. We can use proteins for transporting things in and out. We can use proteins for doing reactions, for receiving information, for recognizing cells, for holding cells together, as well as attaching to other elements such as the extracellular matrix and the cytoskeleton, amongst many, many other protein functions that can be found inside of a cell. Proteins are not random. Cells create the proteins that they need in their environment. They can add and remove proteins as needed. So the mosaic property of a membrane, the many different colors or many different functions that the membrane has, is based upon the many different proteins that the membrane employs. Membrane proteins can either be considered peripheral proteins, which are bound to the surface. So a peripheral protein would either be bound to the outside or the inside. Both of these regions are polar. So a peripheral protein is typically going to be a polar substance, meaning it's got hydrophilic R groups on all of its amino acids. An integral protein is one that's going to penetrate into this hydrophobic core. So at least some region of an integral protein is going to be non-polar. This specific protein is going to have a non-polar region here, and then polar regions here and here. And the polarity of the protein is going to help it orient into the membrane properly. If it spans the entire membrane like this one does, where it goes from the outside all the way to the inside, that's called a transmembrane. Remember, this word trans just means across. So a transmembrane protein is going to be one that goes all the way across the membrane from the inside to the outside. Membrane components are often bonded to carbohydrates. So often you can get lipids that bond with carbohydrates called glycolipids. And then the most common is to have proteins with carbohydrate portions of them called glycoproteins. And these carbohydrate portions are important in helping the cell recognize other cells. So in this example, you can see the way that HIV, which is a virus, which is something we don't want in our cells, actually can get into the cell. So it has to have a CD4 and CCR5, which are both going to be glycoprotein receptors. And it forms this receptor complex here with the spikes on the HIV, and then it allows the HIV virus to be taken into the cell. A person who has a mutation and does not make the CCR5 glycoprotein actually can't be infected by HIV, so they actually become immune because this receptor protein was never formed, and so that HIV is never going to be able to pull into the cell and it gets rejected. So this is an incidence of recognizing something that we don't necessarily want to come in, but we also use glycoproteins for recognizing ourself versus foreign objects like bacteria. Our membrane is a lipid bilayer, right? So we have 2 layers, and you can see they've color-coded. One is orange and one is blue. And each of those layers is made up of 1 layer of phospholipids, and they've got the tails towards each other and then the polar heads out towards the water. Most of the membrane for the cell is made in the rough ER and to some degree the Golgi apparatus as well. So these phospholipid bilayers are produced in here and then transported out to the cell membrane. But look at the sidedness, look at the color coding. Whatever we want to be on the outside of the cell has to start on the inside of the vesicles, because when a vesicle joins with the plasma membrane, the inside gets flipped to the outside of the cell. So the 2 different sides of the membrane are not identical. If I want the cell to be able to recognize another cell, I need to put that glycoprotein or that recognition protein on the outside, which would be kind of these little green Ys. So when I make these proteins, I stick them on the inside of the vesicle so that when the protein eventually gets to the cell membrane, it's actually on the correct outside. So the reason that the 2 sides of the membranes have different structures is because they have different functions. The inside of my cell membrane does not need to recognize other cells because it's not facing other cells. So the structure here implies the function. The main function of our membrane is going to be a selectively permeable barrier. And this is going to be our border control to say who can come in and who cannot come in to the cell, as well as who can leave and who cannot leave the cell. So here's my phospholipid bilayer. So because we have a polar region, a non-polar region, and then another polar region, the hydrophobic or non-polar molecules are able to pass through here fairly easily. So if it's non-polar, it can get through this non-polar region with no issue, just moving down its concentration gradient. So those tend to actually pass through the membrane rapidly. Examples of this would be things like CO2, oxygen gas, which is a good thing. We want to be able to get oxygen into our cells quickly and remove carbon dioxide waste. Hydrophilic molecules, things like ions, calcium, and polar molecules, so things like water, they don't pass easily. Something that is just polar, like water, and still very small, might be able to go through the membrane, but it's not going to do so quickly. Things that are actually charged, like an ion, generally need a protein to help them get through. So those proteins are called transport proteins, and a transport protein specifically moves a specific substance. So you need a sodium ion channel in order to move sodium. You would need a potassium channel in order to move potassium. Transport proteins come in 2 flavors. A channel is kind of like a tunnel, so you can think of it as a tunnel. It doesn't change its shape. It might have a gate on one end that opens and closes, but it generally doesn't change shape. These can be involved in passive transport, where molecules are just moving from one end of the tunnel to the other. So in aquaporin, aqua means water, pore means like pore, a hole. This is just a water channel. So water can get through the membrane, but it's fairly slow. So instead, water can go through an aquaporin much quicker and can move down its concentration gradient faster. A carrier protein is going to change shape. So when it changes shape, it means it's going to have to interact with some molecules. Sometimes it interacts with just the molecule that it's moving, and it can do passive transport. Sometimes the interaction is with ATP, and that's going to result in active transport. So carrier proteins can be found in both passive and active transport. And what this is going to do is the change of shape is going to allow molecules to move across the membrane. So it either needs to bind with something specific to move it across, or it's going to have to be powered by ATP to change shape and move something across the membrane.